Good day, dear colleagues. First of all, I want to thank the organizers for the nice opportunity to participate. It's all the time a great pleasure for me to take part of this event. And actually, the topic I'm presenting today is very new for me. Usually I'm talking about QMMM simulations and so on and enzymatic reactions, but this time it's very different stuff. It's our quite a new project with other members of chemistry department of Moscow State University related to the determination of the aptamer structure of aptamers to spike protein of Sars Cov using nanopore sequencing and molecular modeling. Actually, the first part of my talk will be devoted to the nanopore sequencing. It's quite a new technique. And as for me, it's really a sort of revolution in nanopore, in sequencing, in DNA and RNA sequencing. So the nanopore sequencing is carried out on these small devices. I mean, the size of these devices, like flash memory card, like a huge flash memory card, and it is connected directly to the laptop or computer via conventional USB cable. So all processes occur in this very small cell. And actually it is very complicated, of course, and it has some part where you put your sample. And also the heart of this device is the nanopore. Of course, it can be deduced from the device name. And actually, I will show you how it works and what can be done using this new technique. So the nanopore sequencing works as follows. So this gray huge part is a synthetic membrane. Usually it is made of silicon oxide, but it can be also made of graphene or some other material. And this membrane incorporates transmembrane proteins that can have quite large pores, and DNA or RNA can go through this pore. Here you can see this magenta protein. It's a motor protein that facilitates the conversion of double-strand DNA to single-strand, and it also helps the single-strand DNA to penetrate this pore. So here is the cross-section of the nanopore. So of course, all this happen in solution, and this solution has electrolyte, of course, and the electrolyte is potassium chloride. The idea of this technique is to apply constant potential difference to both sides of this membrane, and of course, potential difference leads to the ionic flow through the membrane through the pores. And of course, positively charged and negatively charged ions go in opposite direction. So this ionic flow can be measured by a specific device incorporated in this nanopore sequencing device. And if you put DNA inside the pore, of course, it blocks some space of the pore cross-section, and the ionic flow, of course, decreases. So the signal, the raw signal that is obtained from this device is ionic current that is measured separately for each pore of this membrane. And as all bases, I mean, there are of course 4 bases, T, C, A, and G. All of them have their own size, and when crossing through the pore, of course, they block different part of the pore, and this results in the in the differences of the degrees of current. So depending on the value of ionic current, you can deduce which particular base was crossing the pore at the moment of the measurement. Importantly, the motor protein that divides double-strand DNA to single-strand DNA works with constant velocity. Therefore, all these horizontal plateaus of ionic current signal are of the same length. Therefore, if you have this time dependence of the ionic current, you can convert it to the sequence of bases. Like here, all, for example, currents that are in red region are imines, then in blue region cytosines, and green and yellow correspond to A and G respectively. So, what are the main applications of this method, and what is its principal difference from the well-known new generation sequencing? First of all, the main strong point is the ability to read long reads. So you can read. Okay. So the main strong point is ability to read long reads of maybe tens, thousands of bases, and the PCR amplification is not required for this method. It's also, of course, really nice. But the drawback or the weak point is that there are quite high errors of sequence determination, but this can be overcome if you just have a large amount of sample DNA and can obtain many, large coverage at each coordinate you're interested in. So the main applications of this method are genome assembly, including de novo genome assembly if you don't have any reference, and also, of course, it is utilized to obtain to find single nucleotide polymorphism and also large rearrangements, like, for example, deletion of entire gene or its duplication or something else. But what we wanted to do in this work is quite a novel application. We wanted to determine the oligonucleotide sequence. Actually, this is very different task because oligonucleotides that are utilized as aptamers are usually short, of course, like tens of bases, and it is how to say, it cannot be directly measured or determined in nanopore experiments, but we utilize some tricks to make long chains and read them. I will show you in my following talk. So then let us move to the Aptamer determination problem. So here is the entire workflow that we utilized in our work. Well, it is quite complicated, and it includes a Celix procedure, then ligation that I told you, that combination of small chains, oligonucleotide chains to long ones, then experiments on determination of the dissociation constant, and the final step is molecular modeling. Actually, the select procedure, I think, is known for more, well, many, it's quite a popular technique nowadays. And it is shortly, it allowed to discriminate uh, prospective compounds from large libraries. So what we did, we had a large library composed of single-strand DNA, oligonucleotides of 71 bases lengths, and we put this mixture with library to the sample containing our target protein. In our case, it was RBD domain of Sars Cov virus. And then, of course, there are some compounds, some oligonucleotides that are bind to the target enzyme, but of course, the majority, does not interact with the target enzyme and can be easily removed from the mixture. Then the oligonucleotides that are bound, so these special experimental procedures utilized to promote dissociation, and already this small amount of oligonucleotides that are prospective from the binding viewpoint are amplified using PCR, and the next cycle is initiated. So usually select procedure is about 10 cycles, so that leads to the enrichment, to the considerable enrichment of the admixture of the sample with the prospective compounds, prospective oligonucleotides. So here is our library. Our Aptamer library was composed of oligonucleotides with the aptamer, so potential aptamer in the middle of this sequence, and constant regions in the left and right flanking regions that were intentionally added so that we can find these middle parts when sequencing. So initially we wanted to have to get aptamers of specific 3D structure. Therefore, we enriched some coordinates of aptamer with either purine or pyrimidine bases to how to say, to promote formation of specific sequences. Well, what we got, I will show you next slides. Actually, it's as usual thing what we want to do and what we obtain are two very different things. So, what about ligation? What about the process of obtaining long reads from these small oligonucleotides? Also, in ideal world, we wanted to get a combination of either primary, forward of aptamers obtained in forward direction, so it's like multiplication of an aptamer, or multiplication of the complementary aptamer, or the combination. And to check whether our ligation indeed resulted in this, we performed nanopore sequencing of the initial library. That we wanted to find enrichment of either purine or pyrimidine bases at certain coordinates, and also the equal contribution of all letters of all bases certain coordinates. So to do this, we performed alignment of left flanking region, and after that, we checked the composition at each coordinate. What we found is that for coordinates that are close to the region that was aligned, we indeed see the considerable enrichment as it should be. But if we go further, we can see that this enrichment decreases, and what was really surprising that the right in the region of right flanking region that is constant for all aptamers we found all this messy stuff, but of course, we expected something like this in left flanking region, but we found these, strange things. At that moment, we understood that actually the ligation process occurs in some different way, and it means that uh, random cleavage of these initial sequences, oligonucleotides happens, and also they are connected in some very specific, very random way. And what we got is a set of very randomly organized uh, long reads. So, but anyway, we had to deal with them. Therefore, we proposed the way to analyze these quite random sequences. So what we did, we utilized alignment frame composed of 9 nucleotides in the beginning of the right flanking region. And then we saw which we checked the enrichment of oligonucleotides at positions that are close to this alignment frame. And then we choose them, and gradually, gradually we moved this alignment frame, and step by step determined the sets of 3 nucleotides of the up. So this allowed us to get the entire sequence of the aptamer. And what is interesting here, you can see that that the majority of columns are colored white, and some others are colored light orange. And the colored columns correspond to the coordinates, to the positions where the letter that was found when sequencing is in line with the expected from from the expected one according to the enrichment that was made in the initial library. So it means that we wanted to have to get the aptamer with specific secondary and 3D structure, but what we get is was very different. Only very small regions are as we expected. So it's not a problem, it's just what we got. And also the interesting thing was that when we tried to determine the aptamer sequence starting from the left side, from the left flanking region, we got much worse results, as the enrichment of the neighboring bases in the beginning of the aptamer was really very small. We thought that maybe the right end of the aptamer is more important for binding, therefore the sequence there is more homogeneous, and maybe the beginning of aptamer does not considerably interact with the target protein, therefore maybe the sequence there is more heterogeneous. So then we chemically synthesized this aptamer and checked its dissociation constant with the RBD domain of SARS-CoV-2 protein or SARS-CoV-2 virus. So we selected the aptamer to the RBD from the Wuhan strain, and when we synthesized it, we got really very low dissociation constant. That is really nice. But when we tried it to Omicron strain, we found no binding. So this means that our aptamer is selective to the Wuhan strain, but not our. So all this experimental data, well, forced us to perform molecular modeling to understand the molecular mechanism of interaction. So to do this, we utilized as an initial point, we used the primary sequence of the Aptamer, and we utilized some web servers like RNA structure, RNA composer, to obtain a 3D structure. And then we performed extended molecular dynamic simulation of this oligonucleotide to obtain the more stable structure. This structure was further utilized in molecular docking. We utilized hot-dog web server to obtain a nucleotide protein complex. And actually, we got, of course, a set of possible complexes, and all of them were utilized for determination of stability of complex by molecular dynamic simulation. So what I mean is that, of course, this hot-dog server is really rapid, but of course, we are not quite confident about the reliability of these results. Therefore, we performed additional MD simulations to find complexes that are more likely to be surprisingly, it turned out that among 13 complexes predicted by Hadoop server, only one was stable in MD simulations. So we utilized this complex as a template to construct the complex with the Omicron, with the RBD domain from Omicron strain, and performed sets of MD simulations for both complexes. And what we found is that the structure of the complex of oligonucleotide of the aptamer with the RBD domain is stable and is really very sort of rigid during simulations, whereas in case of the RBD domain of Omicron strain, the right end does not interact with the protein. Let me remind you that according to our nanopore sequencing data, the right end of the aptamer was much more important as the enrichment of each of the base type at each coordinate is more pronounced at this right end. So of course we tried to obtain some other metrics of these complexes. I mean some quantitative metrics. To do this, we calculated the distribution of number of hydrogen bonds between the protein and oligonucleotide during simulation. And here we can see that the number of hydrogen bonds are much larger in case of the RBD domain from Wuhan strain. That, of course, also explains its so also we perform dynamic network analysis, as in many cases it's an indication of the cooperativity or of motion, and so it also can shed light on what is which complex is more stable. And the left picture demonstrates that there are many relations or connections between protein and aptamer, whereas in in complex with the RBD domain from the Omicron strain, interactions are less pronounced, meaning that the motion is less correlated. I mean, the motion of protein and aptamer is less correlated in case of RBD domain from Omicron. So, I'm moving to conclusions. I demonstrate you how to utilize nanopore sequences in quite new applications of search of aptamer structure. And it's a really long story and huge of course, experimental work and molecular modeling indeed shed light on what is going on the molecular mechanism of interactions and helped to understand the nanopore experiments as well as data on binding constant. And well, of course, I was responsible only for molecular modeling and bioinformatic analysis, whereas the main contributors from the experimental groups are Maria Zvereva and Fyodor Graboyenko. At this point, I'm going to stop, and I will be happy to answer the question.
Thank you very much, Maria. Yeah, I was much surprised because the topic is really far away from your main area of interest, but it was very, very interesting. So we have one question in the chat from Vladimir Sulimov. How do you calculate probability of hydrogen bonds formation?
So we used standard criteria like the distance, distance between heavy atoms like 3.2 angstrom, and also the angle between between hydrogen bond acceptor and H donor group. So between these three atoms to be higher than 120 degrees.
Thank you.
And are there any questions more? I have a question, Maria. What's about debut idea? Why do you expect, there were probably some data showing that RBD domain of the spike protein can be catched, can interact strongly with DNA, RNA, with nucleic acids?
Well, actually, there are already some published data on such interactions, and some aptamers are already suggested. So, and what was the novelty of our project or our work is that, first of all, we wanted to reduce the size of aptamer. Of course, the smaller molecule is, yeah, the better. I mean, the inhibitor. And also, we use this nanopore sequencing to determine the structure of aptamer. But anyway, the idea itself, idea of selection of aptamer to RBD domain is not a new one.
But how selective is such interaction? For instance, the interface, binding interface, does it interfere with ACE2 binding site in RBD, for instance?
Well, depending on the particular sequence of the aptamer, of course, it can be different. For example, there are studies where it was shown that the aptamer also blocks the interaction with AC2, but it binds not to the RBD domain, but to some other. So depending on the particular sequence of the aptamer, the binding site of course will differ. And actually, here I also want to point out that maybe it's the application of such aptamers are not only sort of like potential drugs, but also they can be utilized as in bioanalytical systems to distinguish between different proteins from different strains. So you can just check whether there is interaction or there is no interaction, and at that point you can understand what particular strain was in your sample. I mean, the protein.
Thank you very much. We should switch to the next talk, and there is one more question. See in the chat, please, from Artem Kniga. You can respond in the chat. Thank you very much.
Thank you.
Thank you again.